right. Well, thank you everyone for joining us. I don't know how to make this go away. Thanks everyone for joining us. My name is Annie Carter. I am the Education Programs Manager for Colorado Canyons Association. If you are not familiar with our work, we are essentially the friends group to Colorado's 3 national conservation areas. So we have McInnis Canyons up here outside of Fruta, Dominguez Escalante near Delta, and the Gunnison Gorge National Conservation Area around Delta and Montrose area. CCA works with these 3 communities to engage the public in education, outreach, and stewardship on those 3 landscapes. And our lecture series is a great way to kind of do a deep dive on emerging research and particular topics of interest in those national conservation areas. I had the pleasure of actually going on a river trip this summer with Denita, who is a professor at Colorado Mesa University. And she was sharing with some BLM folks and some funders, and I kind of just got to go along to learn about it. But her really fascinating research on native amphibians and how these invasive bullfrogs are affecting those amphibians. We did a cool night hike up Me Canyon, and it was just an awesome educational experience. I'm sorry you can't all have that tonight, but I think Denita is going to share some great information with us about how you might be able to get involved with her work. So with that, Denita, thanks for joining us tonight. I'm going to turn it over to you, but if anyone has questions, we're going to have some time at the end of the presentation for those. So feel free to put those in the chat or the Q&A, and we will answer those at the end. And with that, I'm going to mute myself. So thank you, Denita.
All right, thank you for having me. I'm excited to share the research that I've been working on with CMU students for the past 5 years, and it has led to some citizen science projects to get volunteers in the community involved in conservation locally. So I'm going to share with you, let me get my presentation up here first. Here we go. Okay, hopefully you can see that all right. So I'm going to share with you the inspiration for these citizen science events. And I've called my citizen science event Bullfrogs for Beers because I thought people needed an incentive to kind of get going and come on out. And it turns out to be a really great incentive. So we'll end the talk with what that event has looked like for the last couple of years. So most of you are probably familiar with the McInnis Canyon NCA, but just in case to kind of orient on the map where we are, I've got just Grand Junction labeled over here. All this green space represents Grand Junction, of course, Palisade to the right, and Fruta up into the northwest there. So the MCNCA, you can see the Colorado River snakes down through here, and the best way to access the canyons that are throughout this NCA are usually by boat. There are quite a few of the canyons that you can hike into. They're very long hikes, but they're not heavily recreated. So they're really great wilderness environments. And then I have a picture down here in the bottom left that kind of shows an overview of an aerial view of what that looks like. So this started when I began my position at CMU. We wanted to know what amphibians are living in the McInnis Canyon NCA. We started to experience things like drought conditions, invasive species that are potential threats to these wilderness environments. We need to know what is there to protect in the first place. So this started a large-scale cooperative agreement. Before I get into that, what that agreement looked like, for those of you that haven't been out into the MCNCA, here's what that habitat looks like generally. So the one on the far left, this is showing the habitat where some of these canyons will drain into the Colorado River. And what you end up seeing is that Colorado River will backwash a little bit up into that drainage, and it creates almost a pond-like environment, which is really nice for amphibians. The middle picture here shows one of the drainages that sometimes you'll see water. It's very ephemeral. So rainfall and snow melt will have a lot of water coming through, but it dries up pretty quickly before the summer hits. And then the picture over to the right, this is showing one of those drainage areas that happen to hold a little puddle of water. And amphibians will utilize those puddles as quickly as they can for breeding. So our primary questions here, getting into the NCA, were which species of amphibians inhabit these canyons? Are non-native bullfrogs also present in the NCA? And also, is there a fungal disease present there? It's a fungal disease that only impacts amphibians. It's called BD for short, and I'll go into a little detail about what that fungal disease looks like. We wanted to know if it was had made it to our amphibians in the MCNCA. So some potential threats to the amphibians of the Western Slope include disease that I mentioned, that BD, and also invasive species. And the American bullfrog is in Western Colorado. It's actually not part of its native range. It was introduced here. And this is an issue because bullfrogs will eat anything that moves. So you can roll a ping pong ball in front of them and they will try to eat it. But that also means that they will try to eat our native species, and they do. They also tend to be reservoirs for certain amphibian diseases. So this means that they can carry this disease with them into these new environments, and they may not experience illness from the disease, but then they can share these puddles with native species who will succumb to that disease. And then the disease I'm mentioning, BD, the long name for it is Batrachochytrium dendrobatidis. And this one has caused global decline of amphibian populations. It's a microscopic fungus. So here I have a picture showing its life cycle. And it's unique in that it is aquatic and microscopic. And it uses chemical cues from frog skin to actually find them in the water. So it swims toward the frog and it actually will embed into their skin layers where it starts to cause complications. And in this image down on the bottom left, this is from a study where they were looking at images of cross sections of amphibian skin that were infected by this fungus. So you can see it almost makes this little vessel, and all those little dots in there are new zoospores or new fungal spores that are about to come out and reinfect new frogs. And so why is it so horrible for amphibians? Their skin is actually really unique and it's extremely important for their overall health. So first thing, amphibians will never drink water. They rely on their water intake entirely from their skin. They have an area called a drink patch on their lower belly and their ventral side. And they will actually go sit in the water and absorb their water that way. But if they're also absorbing this fungus that gets into the skin layers, it starts to interrupt their ability to regulate their water balance. In addition to that, they also take in electrolytes that way. And when this fungus infects the skin, it begins to cause complications with their ability to regulate gas exchange, water balance, and electrolytes. So they experience a couple of different symptoms that I'll show you in the next slide. And on the bottom here, I actually cultured this fungus in the lab to try to better understand it. So I do have a little video, and you can see this one bubble toward the bottom, and there are a lot of little dots inside of that bubble that are moving around. So in that video, those are all zoospores that are ready to come out and complete the life cycle. If an amphibian were to succumb to this infection, the disease is called chytridiomycosis. So the animal starts to act lethargic. They shed their skin excessively, trying to rid themselves of this fungus. Their skin becomes thicker. They have an abnormal posture where they're kind of, you can see the frog in this picture, its nose is pointed downward. They'll stop eating, and eventually it leads to cardiac arrest because of that electrolyte exchange being interrupted. So again, bullfrogs tend to carry this fungus with them, and some of our native species are extremely susceptible to this fungus. So this long-term project started in 2019, and it was a cooperative agreement through Bureau of Land Management, and we've developed partnerships with USGS as well as Colorado Parks and Wildlife. So we have a pretty big team that has been a part of this conservation effort. And it's also allowed for paid undergraduate research experience for 12 CMU students since it started, which is pretty amazing. Here's a couple of my students working in the field. And as I mentioned, access to many of these canyons requires boating down the Colorado River. So in the image to the left are a couple of my students in their own kayaks as we head out for a research trip. And so the McInnis Canyon or Black Ridge Canyon Wilderness area, I included this picture because as you see the Colorado River snaking down, you can see the canyons labeled on here. For example, there's Rattlesnake Canyon, Me Canyon, more. And I'll have a more detailed map of this later with some results. We were trying to sample as many of these canyons as possible in this habitat in the wilderness. We did this through a couple different methods. One of them was through amphibian surveys. Typically at night, we would go out looking for amphibians. They're usually the most active at night, so that's when you can hear them and also find them more easily. If we found them, we would take some information about their GPS coordinates. We would also catch them and take information about their mass and their length. And here are a couple of pictures of students climbing up waterfalls and trying to get to some of these really difficult habitats. The call surveys are a big one, and then the captures as well. And when we would capture them, we actually use sterile cotton swabs. We swab their body, and then we can release them and take that swab back to the lab where CMU students can check to see if they're carrying that fungal disease. So our native amphibians, just in case anyone's not familiar with them, we have 5 of them that we were looking for. So the Woodhouse's toad, this one is pretty common. You might have even seen them in your yard before. They have a fairly large body size for a toad. They have dry, warty skin. And compared to the other toad that's really common, there are pretty big differences that I'll point out to you when we get to that one. The Great Basin spadefoot. This is a toad, but it's really unique. So this one actually has vertical pupils, which is different from the others. And if you were to find one and pick it up, it actually has a spade on the back foot, hence the name spadefoot. And it uses that to dig down deep into the soil to go into kind of a dormancy period when the rains are not there. So it spends about 80% of its life buried, and it comes up after rainfall to breed. This guy over here, the red-spotted toad, they are also pretty common. And hopefully you can see how it got its common name, red-spotted toad. It has those red spots on its back. And in comparison to the Woodhouse's toad, that's also really common, these guys are about half the size. So they're very small. And those red spots usually are a pretty big giveaway. Canyon tree frogs. These are a little more cryptic, kind of like our spadefoots. They're not as easy to see. The best way to find these is a night hike in a canyon near a waterfall. They really like to be in vertical environments, kind of the cliffside or waterfall rock next to a pool of water. That's where they spend most of their time. And lastly, the northern leopard frog. And these ones, they are listed as a state species of concern. And there's some evidence to suggest that the arrival of bullfrogs in this area has led to declines of northern leopard frog populations, possibly for a couple different reasons. First being competition. So they're eating very similar kinds of food sources. Another is going to be the spread of that disease. So the northern leopard frog is actually really susceptible to that BD fungus. And lastly, potentially eating them. So for our non-native amphibian surveys, we went out in a similar fashion. We would do call surveys and we would do a visual encounter survey, which just means that we walk a transect of the canyon and we look for them with headlamps at night. And in this picture here in the middle is one of my students collecting some water samples. You can see that rocky habitat and that drainage that's holding water currently. In addition to the call surveys and the visual surveys, I mentioned that my student was collecting water here. We know that when we go out and do one of these hikes at night, we may miss a bullfrog who might just not be out that night or maybe not in that hour that we were surveying. So another tool that we have to detect species that are maybe there, but we don't see them immediately, it's called environmental DNA or eDNA. And this is where you can collect water samples from streams like this and we filter it in the field. So we're really careful to not allow any contamination. We wear gloves and we filter this water through a small pore filter. And then we can preserve that filter and send it off to a lab for analysis. And when they do these analyses, they are using special markers that look for bullfrog DNA. So we did get quite a few hits from those samples in places where we did not actually see bullfrogs. So this is a map that kind of combines all of the data that we've collected from 2019 to 2022. And I'll walk you through a couple of key points here on this. So we'll start with Devil's Canyon on the right side here. If we look at our little color wheel, we've got orange, we've got Woodhouse's toads, we have Great Basin spadefoot toads, we've got red spotted toads and canyon tree frogs. So Devil's Canyon is actually a place where if you go out in the spring months for a night hike, you can find some of those canyon tree frogs in the waterfall habitats. So that one is heavily recreated. And then as you get farther west into Pollock Canyon and Rattlesnake, some of the regions we sampled, these are not as heavily recreated. So in one of the things that I want to point you to, the jungle green color, the American bullfrog, we find them in Salt Creek. So it's kind of the top middle of the map here. Right underneath that, we found them in Rattlesnake Canyon. We found them in Mee Canyon, Moore Canyon, and McDonald Creek. So most of these canyons look like the initial pictures that I showed you that were, they're very ephemeral. It's mostly rocky and they're going to dry up pretty quickly, except for that area close to the river. But Salt Creek is actually really unique. It is a perennial spring. It runs constantly year round and it's a pretty deep creek. The water that flows out of here comes from larger water bodies upstream. And it's actually used by some fish that are threatened that use that for breeding grounds. So this one is not like the others where it dries up. So that one we expected to find bullfrogs in. And as we come down to Mee Canyon, you'll see that color wheel, we have quite a few species on there. So in addition to bullfrogs, we also found spadefoots. We found tiger salamanders. So that's one that I did not introduce in the native species slide. We found tiger salamanders there, also woodhouses, canyon tree frogs, and red spotted toads. So just about every native organism or every native amphibian except for the northern leopard frog. What I want to mention about the presence of bullfrogs is that in all of these places, except for Salt Creek, so McDonald Creek, Moore Canyon, Rattlesnake, the positives were from eDNA. So this was from a water sample taken close to the river. So we know that in these cases where we find bullfrogs in these canyons, they're usually really close to the Colorado River and they have not moved up into the canyon yet, with the exception of Mee Canyon. Mee Canyon, we've been sampling this one and really zeroing in on trying to sample this one more often because we realized that bullfrogs were able to find habitat and move farther up inside of this canyon. So I'll show you this map next that gives you an idea of where we found bullfrogs exactly and which method we used. So we have that lime green triangle showing eDNA detection. And then the little green circles are showing if we found them visually. So if we look here at Mee Canyon, we zoom in on the left side here, there's a smaller map that shows Mee Canyon. And it shows a couple of different markers for bullfrogs. You notice that there are also little red hash marks in there, and those stand for barriers. If you've ever hiked up into Mee Canyon, there are sections that you come to where you'll have a maybe 20-foot or more cliff where there would be a waterfall, a really big drop off. And we thought that maybe these rocky barriers would keep a bullfrog from making it up to the next level. And unfortunately, we found them above these barriers. So it doesn't appear to be the case that that's keeping them out. We found bullfrogs in this canyon up to 2 miles into Mee Canyon itself. This is the type of barrier that I was talking about. So this is a view from the side of one of those waterfalls. And you can see that there's a little puddle here on the left. And then there's kind of a a big drop off there, and then a puddle at the bottom. And that puddle at the bottom, you can see a little dot there toward the edge of that little pool, and that's actually a bullfrog. And in this year, we found them both at the top of that waterfall and below it. So they're still making it up over these barriers. So where are the bullfrogs coming from? The Colorado River, the areas that I showed you where there's a little bit of backwash from the river, maybe they're breeding there, but I think that it's possible that the upstream of the McInnis Canyon NCA, we have a lot of different water bodies that are in parks that are right near the Colorado River. And many of those water bodies have bullfrogs in them, and very healthy populations of bullfrogs that I've been sampling for a few years. And so on this image, it's showing you Snook's Bottom open space. It's showing you Audubon Nature Preserve in the Connected Lakes area, Corn Lake and Riverside Park, all the way out to Palisade. These are only the 4 that I've had a chance to really get out to with my students, but I know that there are many, many more, including some golf courses and some private ponds that have a huge bullfrog population. So it's important to mention here that there was BD detection in a couple of these parks. So if we see here Audubon Nature Preserve, we've found BD there. And we've also found that it's Snook's Bottom open space. And one thing I forgot to mention, I'm going to go back to this slide here. In 2019, we found that BD fungus in a bullfrog in Mee Canyon. And that was the only BD positive sample that we found throughout all the years of sampling that was in the NCA. But it still is concerning and something that we want to keep monitoring. Coming back here, I thought it's going to take a small army to really understand the impacts these bullfrogs are having, and to try to have any kind of removal effort. So I thought that it would be great to try to involve the public in this effort, more hands on deck. And so citizen science is just that research collaboration between scientists and volunteers, and it really expands the opportunity for data collection that we have. And this data is going to be used for management decisions. So it's we've started to develop a really great database because of these citizen science activities. So the Bullfrogs for Beers was born, and I worked closely with CPW to create marketing materials for the event. We've had quite a few different partners throughout the last few years for these events. So see, we've got volunteers who are recruited through advertisement through Eureka Math and Science Center, using just social media, emails, interested groups like the Audubon Society, Rivers Edge West. We've gotten small grants from Colorado Partners for Amphibian and Reptile Conservation. And I also went out to a couple of breweries in town and explained what we were doing. And Base Camp was the first one to be completely supportive. They donated beers as prizes. So that's where the Bullfrogs for Beers comes in. Volunteers could come out, and they would, when they turned in bullfrogs to a research table, they would be awarded with beer vouchers to go to Base Camp. I've since then, Trail Life this last year jumped on board. They're also donating beers for this effort. And not everybody likes beer, not everybody's 21. So I also partnered with Graf Dairy. They provide some discounted ice cream cones, and also with Pablo's Pizza, they're providing some free slices so that other people that were interested who didn't want beer could partake as well. So families have been coming out and having a great time. So what the event looks like, they would have to do a training over Zoom first. And the training is important so that people who come out to help with this collection understand why we're removing bullfrogs in the population, what impact do they have on our native species, any rules for collection events. So I wanted them to be able to identify the native species as well as the bullfrogs so that we're not grabbing the wrong ones. And also, really importantly, what will happen to the bullfrogs, how they'll be used, how they'll be euthanized, so that they have a very clear expectation about what we're going to be doing there. They also learn things like the fact that we're testing for that fungal disease. So they have to wear gloves between each amphibian that they capture to make sure that we can properly test them for this disease and monitor it. And so the main objectives with the Bullfrogs for Beers was community education, involvement, and removal of invasive species. A lot of people who have gone through the training told me they didn't even realize that bullfrogs were invasive out here, that they might even be an issue. It's created research projects for undergraduate students at CMU. The undergraduate students get to do all of the testing for BD in our lab at CMU. And they also get to assess what these bullfrogs are eating. That is still a big question for us. Are they eating things like our northern leopard frogs? And so they get to do a diet analysis part as well. And then finally, these bullfrogs, I want to get the most use out of them as possible. So they've been donated to classrooms for use in labs. Most classrooms, like scientific classrooms that have to do dissection labs, they'll purchase those frogs from a company. But it makes more sense if we're going to remove an invasive species and it's the same one they would have purchased for a dissection lab. This allows us to really utilize them as much as possible and contribute to an education piece. So this last year, one of our CMU classes used the bullfrogs in the classroom and they did their dissection lab, but they also got to be a part of data collection. When they do their dissections, they're also opening up the stomachs on these bullfrogs and preserving whatever the contents are so that we can collect more data on what they're eating. So the places that we've sampled the most, and I'll go through some data that we've found over the last few years, Snooks Bottom Open Space, Audubon Nature Preserve, and Corn Lake. These have been the ones that we've sampled the most. So we've done it for 3 years now, but this was, I don't have any updated data from 2023 yet. I'm still working on those numbers. But we've developed quite a few partnerships with local conservation groups and businesses as this just keeps growing. The total volunteers are actually over 81. I think we're actually at 100. And total bullfrogs removed is also over 100. So this picture on the bottom left is showing the nature, the Audubon Nature Preserve area. And so this is the kind of setup that I would have at one of these events. I would ask everyone to arrive before nightfall, and they've already completed training, they sign a waiver. And then I have a data table where if they catch a bullfrog, they bring it to me. And if they would like to be part of it, they get to help out with the measuring of the mass and the length, and also swabbing the organisms for BD. And then I keep those bullfrogs and bring them back to CMU. And those bullfrogs are euthanized, but in the most humane way possible. I use a fish anesthetic. So basically just overdose them on a fish anesthetic and they go numb and go to sleep. So that's how they're euthanized ultimately. So 2021, we had 3 training dates and all of them were over Zoom. It seems to be the most accessible for people to just jump on a Zoom call to do their training. And these were the dates that we had. We did Connected Lake Sur, the Audubon Nature Preserve area. We caught 27 bullfrogs. At Snooks Bottom on August 2nd, we caught 16. And then we caught 5 more another night at Snooks Bottom. And for this was our very first event that we put on. And we had some live capture nets that were donated to us by River's Edge West. And the first night of collection, we had 12 volunteers. And I just showed an overview here of the Connected Lakes Audubon area. So you can get an idea. The Colorado River is the one that kind of comes through and looks brown here. And then Connected Lakes has all of these water bodies right there next to that Colorado River. And they're just full of bullfrogs. So it's really easy access for these bullfrogs that are potentially source populations to get into that Colorado River and make it downstream. Interestingly, for this first one, we found of the 27 bullfrogs, most of them were juveniles. They were not even adults. So that was an interesting finding that we keep seeing at that particular site. Fortunately, we did not find any BD within any of those amphibians. At Snooks Bottom, we had 2 nights of collection with 12 volunteers on both nights. And to show you that aerial view, again, you can see the Colorado River coming through here in that top right picture. And you have Snooks Bottom open space on the left. And even on the right, I have another water body I need to start sampling. Those are just bordering the river on each side. We found no BD at that site either. For 2022, we also had 3 training dates over Zoom. So we went back to Snooks Bottom again and found 10 bullfrogs. And then Connected Lakes, we found 19. And then for the first time, I sampled at Corn Lake, and a Girl Scout troop came out to do the sampling, and they found 14 bullfrogs. For the Snooks Bottom site, we found 10 bullfrogs. All of them were large adults. Tends to be a thing that we find at Snooks Bottom are very large adults versus Connected Lakes and Audubon Nature Preserve area. We find mostly juveniles. For this 2022 sampling, we did not find any BD. And at the Audubon Nature Preserve area, we had 19 bullfrogs that time, mostly juveniles again. Fortunately, we did not find any BD. And then at Corn Lake, those Girl Scouts came out. There were actually 21 of them in total with parents and all the Girl Scouts. And they found 14 bullfrogs. And it was a mix of adults and juveniles. And we did not find BD in that site either. So before I go on to the next slide, I don't have all of the data figured out yet for my 2023 year. However, we did find BD positive bullfrog at this Audubon Nature Preserve site. And we found that before in 2019. So it's been a couple of years, but we keep seeing the disease pop up in low numbers. So the vertebrate diet contents, we've had CMU students working on figuring out what they're eating. And this was from some bullfrogs that were captured by volunteers. For the Connected Lakes area, we found that they were eating fish. So that top picture on the right, it's a partially digested fish, but there were components of it that we could figure out it was a fish. We just didn't know what kind. And then on the bottom, you can see that that's a toad. So that's actually a Woodhouse's toad that this bullfrog had eaten. And then on the bottom left, this is a pelvis of another frog. We don't know what kind. It was too far digested, but we could tell that it was a pretty sizable frog that had been eaten. And then the Girl Scouts, from the trip that they went out and captured, they found a bullfrog that had eaten a snake. It actually had eaten a juvenile garter snake that was pretty large compared to the bullfrog itself. And we continue to analyze these diet contents every time we get more bullfrogs come in from these events. And it's significant because it gives us evidence that they are consuming our native vertebrates. They're consuming snakes, they're consuming amphibians, and also we've even found rodents and fish. And this helps us support grants for continued research and also helps with management decisions. So future directions for this, I will keep doing bullfrogs for beers as long as volunteers are interested in coming out and learning about this and helping out. So I'd like to include more locations and more events, expanding partnerships to continue the momentum that we have. And one of those, in case you're interested in this, there's a group called CCAST. They're Collaborative Conservation Adaptive Strategy Toolbox. And this is a group that has come together and they have webinars and they discuss case studies on bullfrog removal and additional native, non-native species removal all over the Western United States. So they've had a couple of success stories on bullfrog removal that they've cataloged there. I would love to include river boaters. I think that some of these canyons that we know bullfrogs are found in, if we could hold some of these volunteer events in those canyons, or even have trained boaters that would be willing and interested to collect bullfrogs and then bring them back to the takeout, and they would be helping to remove those bullfrogs from the habitat. And then finally, I mentioned earlier that Mee Canyon, we really kind of focused in on Mee Canyon because we found bullfrogs there quite a bit up into the canyon, and one of them had had BD. So we've really focused on that one as kind of a continued research based on that habitat being able to provide provides habitat for a lot of different native species, but also these bullfrogs. So we've purchased remote acoustic and water detection devices. And what this allows us to do is just place these detection devices out there. And so if water shows up in the canyon in the spring, I'll know how much water is there, what the water temperature is, and the acoustic devices paired with that will tell me when the frogs begin to call. And each frog species and toad species have their own characteristic call. So just based on that acoustic data, I can tell who is there. So we'll be able to hear if bullfrogs are showing up at certain times a year only. And it'd be really great to get volunteers involved, people who do hike through Me Canyon or go out for boating. They can go and download this data through Bluetooth, which would be really helpful. And I am happy to take any questions that you guys have.
Well, thank you, Denita. Sorry, I'm just getting things squared away. That was so cool. I, learned so much from you on our trip, but this was really nice to see the data and your methods and everything. I can't wait to participate in Bullfrogs for Beers. So folks, if you have questions, please send them in chat or Q&A. While I'm getting some questions in, I just want to share with you, Denita, some of the comments that came in while you were speaking. So Terry said when she saw a northern leopard frog, CPW biologist Dan Newbaum told her that you must be doing a good job on bullfrogs in the Connected Lakes area. So commandments there. We also have Brett says, beers and root beers for bullfrogs with the kids was so much fun and more than a fair trade. Kara says, I love that you're sharing how you are compassionately euthanizing the frogs as opposed to some other efforts like squish the lantern bugs and culling barred owls. So yeah, you're getting a lot of enthusiasm in the chat. But we have some great questions so far. So Luke wants to know, any evidence of BD transport from one habitat to another via humans or dogs? If so, are there any recommendations on how to decontaminate?
That's a really good question. There's no evidence right now that humans or dogs would transport them. We follow decontamination protocols for any boots or shoes or waders that have been in an aquatic environment because there are other pathogens that can easily travel on those. So we usually use, there's a quat sanitizer solution that CPW recommends. It can, if it stays wet, it can potentially be transferred. BD does not do very well once it's dried out. So for that particular pathogen, it's not going to be easily transported as long as it's dried out on those waders. But there are other things that can affect fish if you go from one aquatic environment to another. Yeah.
That was a great question. We have another one. Brett would like to know, are there bullfrogs on Pinyon Mesa? Could they possibly be moving downstream rather than upstream?
I don't know. I've never checked. So Pinyon Mesa, I'm actually not entirely familiar where that is, but I would like to check it out. I know that there are some areas in Glade Park, for example, and we wondered if bullfrogs are being washed down into the NCA from like cattle ponds and private ponds that are upstream there. So something like checking out Pinyon Mesa, I have not done yet, but I will add it to my list. I would love to.
Yeah, Pinyon Mesa is in that Glade Park area. So I think same thought process that you're having. Yeah, thanks, Brett. Okay, let's see. So we definitely have some interest in volunteering and, an email list or anything. I also want to add, Denita, I think I'll be sending you an email later. CCA would love to get more involved and mobilize our volunteer base. But we do have some interest amongst participants about an email list to join, especially in boater volunteer options.
Yeah. So this next year, I really like to test out the boater volunteer option. I'd like to actually run a Bullfrogs for Beers event at the Me Canyon campsite. That's something that I'm trying to arrange with Bureau of Land Management. But as far as an email list, I usually send a flyer out to any groups like Rivers Edge West or to you guys or Eureka, and I can do that going forward. I need to develop an email list because I have a lot of volunteers who would like to be a part of this. So I'll start by sending flyers to you guys, and then maybe that's an easy way to distribute that.
Perfect. Yeah. I also can put the plug in. There is a growing collaborative effort amongst nonprofits and folks that engage volunteers in the area, the West Slope Outdoor Volunteers. And that is almost every group you listed and some others as well, to just share volunteer events and share those volunteers between organizations. And so that can be another great place to post as well.
Great.
Scott would like to know, what months are you having the best success on finding bullfrogs? And what months are you running your events?
That's a great question because my first 2 years, based on my other research in the canyon, I didn't have time to run the events until July and early August. And this last year, I decided to try to run the volunteer events in May. And it was surprisingly still too cold for some of the bullfrogs. It seems like July and like all the way through July and early August are the best time to find these bullfrogs in the habitats that we're looking. So I'll probably continue to do those during like the late summer.
Very cool. Emily would like to know, and she's actually a former colleague, have you experimented at all with eDNA sampling for BD in addition to swabbing frogs? We at BLM were considering sampling for BD this way, but have not started yet. Curious if sampling water rather than amphibians is not targeted enough.
I think that's been a question that some other researchers have tried to tackle. Can we actually use eDNA to find BD? And I think the trouble is that the amount of BD that you might get is not enough. It's not like a bullfrog sitting in water is shedding a lot of DNA. You would have to get a single zoospore at least. So I think that it seems like it's not concentrated enough in the water that we're collecting to really get a good sample to detect it.
Yeah. Folks, please feel free to add in some more questions. I had one. It seemed like in a lot of your events, you were finding mostly juveniles. Do you think there's a reason for that?
So in the events at Audubon Nature Preserve, they're mostly juveniles. And then at places like Snook's Bottom, we find some of the biggest bullfrogs I've ever seen. There's a very different insect prey profile at each of those sites. So Snook's Bottom has very different kinds of food than you'd find at Audubon Nature Preserve. But I have not figured out yet why there are more juveniles in this place and not the other. It's an open question right now.
My thought was maybe they were just easier to catch, less experience in life and, a little, a little slower. It looks like, okay, here's another question. Do you think there is a place for outreach to pet stores? I've noticed many in the area sell bullfrog tadpoles, and I wonder how many get released since they get so big.
I've also seen them in pet stores, and I don't know how to approach that one, but maybe it's worth reaching out and having a discussion to see if that's something that that they know that they're invasive here and that they're actually an issue, because maybe they don't. A lot of people don't even realize that they are invasive in this area. So that's a starting point, is maybe I could start having conversations with those pet stores, but I have not done so yet. I do know that some people will use bullfrog tadpoles for bait for fishing. And maybe that's one reason that they still get kind of transported and sold in some places, including like bait shops.
Yeah. Well, we can give it another minute to see if some more. Oh, here's another one. Is there any effort to encourage anglers to help control bullfrogs separately from your Bullfrogs for Beers efforts, or is it like bailing the ocean?
Um, so I want to start doing some more concentrated like survey efforts to see if we're seeing a difference in leopard frog populations and bullfrog populations at sites where I know that they both exist together. It seems like there's some anecdotal evidence that removal of bullfrogs, we're starting to see more leopard frogs in some of these places. But I don't have numbers to back that up yet. I think that every bullfrog removal is helpful. People have different methods for doing it. If you have a fishing permit, you have an unlimited bag limit on how many bullfrogs you can capture, but you cannot transport live animals. So that is one piece to consider. So someone would have to use whatever euthanasia effort that they would like to. But I think that removal of every bullfrog is helpful because a single bullfrog lays hundreds of eggs. And bullfrog tadpoles can stay in an aquatic environment as tadpoles for years. As long as the conditions are suitable, they will just stay there for a very long time. So you sometimes don't even know how extensive, like how dense that population of bullfrogs is, because we see the juveniles and we see the adults, but we may not see those tadpoles that are also in that pond or that lake.
Yeah. Um, Yeah, kind of like a seed bank of weeds is a comparison, and some more folks thinking the Audubon site is so close to Grand Junction, wondering if that pet trade might be a source of the high number of young frogs. So yeah, that's a complex issue, and I think the community outreach piece of getting more folks involved in the volunteer efforts and potentially finding those unique ways to communicate could be beneficial. These are all some really good ideas from people on the lecture. Um, we still have some time for questions. We can see if any more come in, but I think I'll use this opportunity to just plug CCA a little bit. If you enjoy these lectures and find a benefit from them, please consider checking out our website, becoming a member. There's still time in the year to make a year-end gift, and we really just love being able to share this research with folks and get people involved in all kinds of topics on our national conservation areas. Like I said, Denita, we would really love to get more involved in your work and that, education piece, the restoration and volunteer aspect of it too. So we can talk off the lecture. But thank you for, sharing this with our community. I think it's really important to understand the problem and to all think about how we can be contributing to it or helping solve it. So I really appreciate your time. And I think with that, we will wrap up for the night. Thanks so much, Denita. Keep up the great conservation work is in the chat. So thank you all for being here tonight. You'll get an email from me tomorrow with the recording link. And if there are any links that you'd like to have included in that as well, Denita, I can have that in the email to everyone.
Okay, that sounds great. Thank you for having me.
All right. Thank you all. Happy holidays and enjoy the rest of your Tuesday evening.